This is lecture 19 in Principles of Metabolism. This is the last lecture. And in this lecture, we'll look at some genomic aspects of metabolism. So let's look at the overall situation of metabolism from the genomic perspective. So looking at the human genome, after the sequencing of the human genome, we have a reasonably good idea about how many metabolic enzymes there are to start with. This depends a bit on how what you consider a metabolic enzyme. So I would say there is about 1,500 to 2,000 genes that encode metabolic enzymes. But what do you include in that class? So I tend to exclude things which are kinases acting on proteins, but also lipid metabolism genes that are part of signaling. There's a lot of phospholipid signaling enzymes and things like that, which are in a way enzymes, but not really doing metabolism. But you can draw these boundaries a little differently, it depends on who you ask. But I would say 1,500 to 2,000 genes. And add to that, there's a few hundred transporters that we saw in the previous lecture, which encode these transporters that move between different compartments. And then on top of this, there's hundreds of enzyme-like kind of genes. And this is even more uncertain. You will definitely find a lot of gene names that are called enzyme something like. And those are similar by structure. We don't know if they're actually enzymes. They haven't been tested, but they could be. So that's sort of the white spots on the map. But roughly these are the type of numbers we're dealing with. And when it comes to characterizing enzymes, it's the case that the enzymes in central metabolism are most well known, and that's perhaps not so strange. This is what we have been studying the most. So if you think about the bow tie model again that we had in the introduction lecture, at the center where you have the major reactions that carry a lot of material and that occur in all cell types, these are easiest to study, they are easiest to find and express and so on. So I would say about 80% of these enzymes have been found. If you go to the periphery, to these sides on the input sort of processing side and the output sides, this number 80% that drops very quickly. So some specialized branches of metabolism, certain branches of lipid metabolism, for example, is very sort of poorly charted. We don't know what enzymes there are, and we don't know what genes encode some metabolic activities that we have seen. So this varies a lot depending on what sort of subsystem of metabolism you look at. To the right here, you see a graph from a review sort of synthesis by Dennis and Whitcup. So they did a bit of statistics and tried to figure out from the metabolic activities that we know of that have been categorized as easy numbers, this classification scheme. If we look at various organisms, they looked at human, but also other things, what fraction of those now have a known enzyme, so an encoding gene? And so this varies over time, and it goes both up and down because we discover more and more genes, but we also discover more and more easy numbers. And at present day, it looks like about 70%, maybe towards 80 now. This paper is a bit old. Say 80% of all the easy numbers have been identified. We have genes for those, and the rest are orphan. So quite a lot, I would say. I mean, metabolism is comparatively well charted. If you compare it to some signaling pathways where we know like a few percent, this is comparatively good, but there are still blank spots. Now, when looking at genes encoding enzymes, I think one fascinating aspect is the idea of isozymes. So this is isoforms of enzymes. This was discovered in the late 50s. As everything in biochemistry, the original references are old. So PNAS paper back then. And so what people saw is that when you ran these extracts on gels and looked for activities, you found multiple bands that had a particular activity. So there were multiple proteins encoding a single metabolic activity. single reaction. And this can happen in many ways. Of course, today we know a lot more about how this happens. There are many examples of paralogous genes. So you have two distinct genes that encode proteins that have the same enzymatic activity. There are often gene duplications that have later on specialized and sort of gained specific properties. And then there's a lot of splice variants, alternative transcripts of various forms, or translation variants that somehow yield different isozymes from a single gene. So this is interesting, I think, because isozymes tend to vary in subtle ways. So there are many cases where specific isozymes are expressed in particular conditions. This has been studied a lot in cancer metabolism, which my lab works in. And it was early on recognized that when you looked at embryonic type of tissues and compared that to adult tissues, or compare that to neoplasms, to cancer growth, it was clear that there were many metabolic activities that differed between these, sort of switched back and forth depending on going from normal to embryonic to cancer. And then looking at the isozymes they knew at the time, they recognized pretty quickly that there are these dramatic shifts of, in this case, an isozyme in glycolysis, where normal and adult tissues might express completely different isoforms compared to the fetal stages. There's usually this progression that an early fetal stage is extreme, and then a late is intermediate, and then you get to birth. And then you see a corresponding pattern sometimes showing up in cancers, and in this case hepatomas. So that a well-differentiated tumor that is sort of normal almost in histology looks like the normal case in its isoform expression. Something that is somewhere in between in differentiation, well differentiated, is in between in isozyme expression. And then poorly differentiated tumors look like the early fetal tissue. So these type of patterns have been seen for a while. This indicates that isozymes really have specialized functions. So we don't know really how many isozymes there are, and one reason is that even though we could look at the genome and predict splice variants and things like that, there are also many cases where on the actual enzyme level, tertiary structure, the enzymes are formed by multimers of subunits. And in many cases, you can then have this kind of combinatorial phenomena. So you might have two paralogous genes, A and B, but if your enzyme is a tetramer, then you could have all the possible combinations giving rise to different isozymes. And this actually happens. So lactate dehydrogenase is one well-known example of this. And so there's two major forms that are expressed, two paralogous genes that encode these forms. But it's a tetramer, and so you can form at least five. If you just count the number of A's and B's, then you can form 5 variants of lactate dehydrogenase. You could argue that it might also matter in which position A and B is in this tetramer, but usually people talk about the 5 possible forms. And these differ by mass, then, so you can run them on various gels. And you can indeed see that there are differences in all of these 5 forms across tissues and across embryonic development. So this is highly regulated, obviously, and it is clear that you can generate a great variety of isozyme forms with perhaps different kinetic properties or different regulatory properties from distinct genes. So what do all these forms of enzymes do, and why do we have so many versions of everything? So it's clear if you start looking at the expression patterns of these across tissues and cell types that there is a lot of specialization. So on the left you see a heat map of expression at the mRNA level for about 1,000 metabolic enzymes across a collection of immune cell types. So this is, of course, a particular branch of cell development and only a particular type of tissues. But already within immune cell types, which is a smaller collection, you still see a lot of functional specialization. So you see this type of high red blocks of high expression in particular types or clades of immune cells. You have particular enzyme systems being expressed. So this suggests that there is definitely some kind of coordinated function of this. If you look at genes a bit more in detail, if you look at isoforms of a particular enzyme across tissues, like to the right is an example of aldolase, you realize that there are specific versions of enzymes that specialize in particular tissues. So aldolase, for example, there is a kind of a housekeeping, if you want to call it, type of isoform that is expressed in a lot of tissues, in some higher than others and not present in all, but kind of broad expression pattern. Then there's a very restricted tissue-specific form, the B form, which is present in liver and kidney and intestine. You can't see these labels, of course, but you just have to take my word for it as a liver form. And aldolase C is present in a lot of neuronal cells, so this is usually called the brain form. And obviously it has some special function there. And we think that for the most part, these isoforms do carry out the same reaction. They might differ a bit in cofactor specificity or requirements for ions or counter ions or things like that. But by and large, they are the same enzymes. So most likely what is going on is that throughout evolution, these have acquired different regulatory domains. So they might have a bit different Km, Michaelis-Menten constants, a bit different kinetics, but they are probably also just regulated differently. So they might have acquired binding sites for being phosphorylated or modified somehow, and so that they can be controlled in different ways in different tissues. And in case you think that these expression patterns could just be artifacts or have nothing to do with metabolism in some way, you can also look at metabolite content in tissues. So to the right, there is an example from our lab. This data set was never published, but this is a set of 15 different mouse tissues in a few replicates each, and a couple of 100 polar metabolites that vary across these tissues. And again, you see this type of checkerboard patterns in heat maps. So you have these blocks of high abundance of particular metabolites or classes of metabolites in specific tissues. And a lot of the actual tissue specificity here has been described at some point in the literature, but it's spread across hundreds of articles. But there are quite a few known sort of tissue-specific metabolites. These are things like amino acids, particular forms of amino acids that seem to be stored in particular tissues. Citrulline, for example, is very high in the intestine for some reason, and many other things that are tissue specific. So it really seems like the variations we see in enzyme expression patterns across tissues also translates to metabolic specialization. This is still very poorly understood overall how this works, but it's clear that there are different types of metabolisms in different tissues. So this is a highly specialized process. In some organisms, genes are organized structurally in the genome so that metabolic pathways are encoded by nearby genes and they are sort of expressed at one time. And this is most clear in bacteria, where you have these operons that transcribe and express a whole sequence of different genes at a time. And these are often metabolic pathways. So if you study microbiology, you will see a lot of these. And it's a really sophisticated organization where you can basically transcribe an entire pathway at a time. People have looked for similar things in eukaryotes, in higher eukaryotes, and even in humans, but this type of organization doesn't seem to be present. There are cases where you have one or a couple of related enzymes, or an enzyme and a transporter or something like that, co-localized in the genome. But by and large, this doesn't happen so much. What does happen in higher eukaryotes is that related enzymatic activities tend to be fused into multi-domain proteins. So these there are quite a few examples of. So one example shown here on the bottom left is the multifunctional enzyme in pyrimidine synthesis. So this is carbamyl phosphate synthase, and there's an aspartate metabolizing enzyme that adds aspartate to the pyrimidine ring, and then there is dihydroorotate that finalizes this pyrimidine ring and makes orotate. So these three initial enzymatic steps in the pyrimidine synthesis pathway are encoded by the same gene. It has three distinct domains. And you can see that each of these domains correspond to individual genes in prokaryotes and lower organisms. So this is clearly three different proteins that have fused in evolution. So that instead of these operons, we tend to have single genes, but which are encode multi-domain proteins. Another kind of famous example of this is fatty acid synthase. So this is also a multi-domain protein. We looked a bit at fatty acid synthesis in the lipid lecture. And as you recall, there are several steps. There is elongation. There is these steps where you have to reduce the growing fatty acid chain. A couple of steps, you get rid of oxygen. And these steps are located on different domains on this synthase enzyme. It's a really nice piece of structural biology. So you have a part of the enzyme holding on to the growing fatty acids, and it's kind of swinging between these domains. These enzymatic domains carry out different functions on the elongating fatty acid. So this is a structural picture from a PNAS paper on this enzyme. So this is another important organizing principle for metabolism.